para iniciar a apresentação da conferencista de hoje, né? Então, somente para lembrar, então, principalmente, né, os alunos de graduação e pós-graduação que estão interessados em solicitar os créditos, né, ou que estão inscritos na disciplina da graduação, que, então, a conferência de hoje está valendo para esses créditos, né, de quarta-feira, mas vocês precisam estar logados aqui pelo Google Meet. E, bom, além disso, então, esse simpósio de biologia celular, microscopia e parasitologia, ele continua ainda semana que vem também, até a sexta-feira da semana que vem. Lembrando que essas outras conferências do simpósio, que não são as de quarta-feira, não estão valendo os créditos especificamente para essa disciplina da graduação e também para da pós, né, essa outra disciplina da pós relacionada aos seminários, mas que eu gostaria também de agradecer também ao programa de parasitologia por essas 3 conferências, que as 2 primeiras já foram excelentes, acredito que a de hoje também tenho certeza que será, e agradecer pela indicação e pela organização dessas conferências, e convidar a todos também para a semana que vem, na próxima quarta-feira, nós teremos a continuidade do ciclo de conferências com o professor João Viola. Nós teremos algumas conferências ainda, se eu não me engano, mais 2 depois do João Viola, antes de voltar a ter um programa como convidado, que seria justamente o programa de biologia molecular. Mas essa programação a gente vai passando por e-mail, mas para a semana que vem já estão todos convidados para a conferência do professor João Viola. Então, obrigado a todos, a todas, e vou passar então a palavra para a professora Suzana.
Obrigada, Pedro. Profesora, cuando la señora quiera, puede darle al botoncito. Erika, ¿todo cierto?
Tudo certo.
Tá bom. Dear Guadalupe, thank you very much to be with us today. In the name of all the students, it's a pleasure to have you with us today and have the pleasure of hearing about your excellent work and the perspective that you have of our students and our science. And thank you very much to be with us today. Es un placer enorme tener a la doctora con nosotros. Muchísimas gracias por estar con nosotros. Con certeza su charla va a ser espectacular. Por los 2 minutitos antes que conseguimos conversar, yo creo que la inteligencia, la idea, el corazón que la señora tiene para los alumnos y para cómo enfrentar las situaciones de crisis y proteger el futuro de la ciencia ya muestra la grande científica que usted es. Entonces, en mi corazón usted conquistó con esa frase que espero que repita para que todos nuestros alumnos puedan escuchar. Claro. Muchísimo obrigada en nombre del Instituto de Biofísica Carlos Chagas Filho por estar con nosotros hoy. Profesor Vanderlei.
Ok. Palabras de una española para una mexicana.
Thank you. Thank you very much.
Good morning, everyone. It's a great pleasure for me to introduce Professor Guadalupe Ortega-Perries, a good friend and one of the most distinguished parasitologists in Latin America. She made her basic formation at the famous Universidad Nacional Autónoma de México, probably one of the largest universities in the world. I always remember my visits to that university. We have more than 400,000 students. I don't know the numbers today, but she made her master degrees at the Faculty of Science of UNAM, and then went to England, where she got the PhD at the University of Bristol. And in did Guadalupe stayed in England for several years in different times. She did the postdoc at the National Institute of Medical Research in Mill Hill, London, when visiting scientist at the Harvard School of Public Health, at the Central Laboratory of Veterinary Research and Parasitology in France, Department of Veterinary Pathobiology at College of Veterinary Medicine and Biomedical Science, Investigacyon, the Centro de Investigación y Estudios Avanzados de México, one institutions where I had the opportunity to work for several times during several years with Adolfo Martinez Palomo, that will also give a conference for us next week. She's at the Department of Genetics and Molecular Biology, is full professor, was head of this department for several years. She's member of the editorial board of very important parasitology journals, such as the Advances in Parasitology, the Journal of Infection in Developed Countries, the Parasitology from London, and very recent, she was the editor of two issues of the Advances in Parasitology dealing with Giardia and Giardias. He did two excellent publications where you can find the most recent data on Giardia and Giardias. She is a very enthusiastic organizer of meetings. I did have the opportunity to visit to participate of the Giardia meeting in Morelia, but Marlene Benchimol did, and Guadalupe is participating of the organization of these meetings, independently of in which takes place. She is a member of the Mexican Academy of Science. And today we will talk about Giardia and Giardias. But her work, it's mainly focused on pathogenesis, parasite epithelial cell interaction, virulence factors, susceptibility of parasites to drugs, mechanisms involving drug resistance, and so on. So that's Guadalupe. It's really a great pleasure for us to have you here. Please, you can start your lecture.
Thank you very much, Wanderly. It's a really very nice introduction. And I really would like to thank you for inviting me to participate in this symposium and to have the opportunity to share this platform with such an enormous amount of very important speakers. And what I thought for my presentation is to prepare a talk that will actually help and show the students particularly that depending on the way that you approach your project, the things that you want to do in research, you can do it. And then you can actually use many tools to achieve the goals that you put. But it's very important that you think the way that things can be done. And I'm sure the students remember one of the speakers that mentioned that is thinking about what you want to do and how to approach it. And I'm sure the students have this energy, that possibility, and that is my main goal for them today, that I can actually show you how the preparation of a project and how to reach or to answer the questions that you have. So I hope that the people that are listening to me understand that most of my efforts today will be devoted to the students. So I'm going to be talking, as Professor Wanderley mentioned, about the role of virulent factors, of Giardia virulent factors, in the host-parasite relationship. Well, Giardia duodenalis life cycle, as you know, is a direct cycle, and Giardia duodenalis is a protozoan parasite that infects the host gastrointestinal tract, causing the disease known as giardiasis. And how does the cycle initiates? Well, cysts, which are the infected stage of the parasite, and the trophozoite, which is the vegetative stage, are the two entities of the cycle. And the cysts are the ones that contaminate water or food. And once the host ingests the cyst, it goes through the intestinal tract and then initiate the excission procedure in the stomach. Then out from each cyst, there's two trophozoites that emerge and then divide by fission binary, and then they will colonize the upper part of the intestine. Once they are there, that is where the pathology develops, because there is an interaction of the trophozoites with the intestinal epithelium. And if the conditions of peristalsis and that they are not favorable for the parasite, then it goes and moves into the colon, and eventually where the encystation procedure starts again, and then the cyst can be expelled and into the environment. They may not survive for a long time, but they will contaminate while they are still infective. They contaminate water and food, and then the cycle starts. So, epidemiology. Epidemiology is quite important. It's giardiasis is disease which is worldwide distributed, and in a recent report they mentioned that there's 20.2 million cases per year, and you can see the distribution around here with the developed countries, developing countries, and in fact there is a high prevalence of this infection in developing countries. It mainly affects children between 1 to 5 years of age, in which they cause a really severe acute diseases. Dogs and cats can be synoptic source of infection for humans. And one very important thing that I will refer briefly in my presentation is that this disease has been related to post-infection sequelae such as colitis, irritable bowel syndrome, and chronic bendazole, nitazoxanide, furazolidone, and paramomycin. Giardia duodenalis in particular, which is also called Giardia lamblia and Giardia intestinalis, has been classified in 8 different genetic subtypes, named H to H, and A and B are the ones that mainly infect humans. The Giardia, the clinical spectrum of Giardia infection goes from asymptomatic cases, which are the most common ones in general, according to the different regions, but then you can have the acute giardiasis, which is it induces diarrhea, but eventually it gets protected. So there is a response from the host that actually controls it. But you can also have persistent diarrhea, which is called the chronic giardiasis, when there is malabsorption, and of course there's development and enteropathology, which actually brings us to very severe cases in some patients. So what are the determinants of the disease that are present? Is one of them is the Giardia strain, as I mentioned, the source of nutrients, and virulence factors, which I'm going to refer to today, just the ones that we have studied. Co-enteropathogens and microbiota, very important. I also will mention that briefly. And host genetics and immunity. Also, immune modulation by Giardia is important. So, as I mentioned before, there are different chronic sequelae that I just show them here, which are being related to the giardiasis. So in the pathology of giardiasis, the attachment of trophozoites to the intestinal epithelial cells is the first step in pathogenesis, is essential for the colonization of the host, and is a primary cause of physiological dysfunction in epithelial cells. And some of the people that work, I think we have we have achieved an agreement that this is a multifactorial process. So, what are the factors that participate in the addition of the parasite to the epithelial cells? Is the trophozoites structures, which include the adhesive. This is, in fact, one of the major features in Giardia, the ventrolateral fringe, the lateral ridge as well. And the mechanisms that have been proposed are hydrodynamic forces and regulation of contractile activity by the cytoskeleton. Also, they are, and that's what we have become quite interested in, non-metabolic proteins and metabolic proteins, some of which I'm going to mention in next. So, what several groups have already studied is that in the interaction of Giardia with epithelial cells releases toxin-like molecules, energy metabolism enzymes, agenolytic like glycolytic, like ornithine, carbamyl transferase, enolase, and arginine deaminase, and non-metabolic proteins. Some of them are the cysteine-rich surface proteins, high cysteine membrane proteins, and variant surface proteins. I'm going to refer as BSP-B10A is the one that we have studied. But also, and very importantly, are the proteolytic enzymes, among which is giardipine. And more recently, tenasins also have been reported to be secreted by the when Giardia interacts with the epithelial cells. And in a very nice work by Emery, Corbin, and colleagues, they looked by the differential expression of genes during the interaction of trophocytes with epithelial cells. And if you look in here, the Giardia differential expressions are, among them are the BSPs and cysteine proteases, which are the ones that I'm going to refer to. Also, there are others that are downregulated, and in the epithelial cells, the cytokines, RNA binding proteins, tight and adherent junction genes, as well as some DNA replication and chemokines that seem to be downregulated. So, considering the our interest to identify, analyze, and characterize the proteases, this work was done quite some time ago, but it was a pioneer work in terms of defining which are the proteins that are secreted and have a proteolytic activity. What we did then was to use the trophozoites and that were interacting with the X cells, which is epithelial cells from the ileum of rats. And what when we looked, then we saw that there were several protein bands with proteolytic activity. However, if we use an inhibitor named TPCK or E64, we then find that the proteolytic activity decreases quite a lot. So then that gives us an idea that some of these secreted proteases were cysteine proteases. So in the future, and I think a lot of the people or most of the people that work now with secreted proteases from this parasite are using these inhibitors. So this pioneer work prompted us to define which are these proteins. How did we approach this? And that is important that we are not using too sophisticated methods. We use monoclonal antibodies that a lot of people, and they're the surface of the trophozoites as well as the flagella. When we purify this antibody, what we saw in a Western blot analysis, that a protein with approximately 25 kilodalton protein was recognized by this monoclonal antibody, and that also, if we run the gel in non-reducing conditions, we can see that is also in a high molecular weight complex. If we looked at the proteolytic activity, we then found that indeed this protein has a proteolytic activity. This is a cymogram analysis, and we use another protease inhibitor, CA074. As you can see, there is no that inhibits the activity of this protein. This is the actual data on the mass spectrometry analysis, in which this I'm just showing here the diagram of the mature protein, which has got 25 kilodalton protein. And when we did that, we indeed find that this protein is a catechin-like protease. And as you can see here, the activity of the protein is marked in a circle, and also in a dot plot in the 2D gels. So based on the characteristics of this particular protein, we decided to name it Giardia, because it has got characteristics of the papain-like family. So we had the monoclonal antibody, then what could we do in terms of looking for the interaction of the phospholipids that are expressing this protein? So what we did was a very straightforward experiment in which we label the parasite. Then we interact the trophosites with the epithelial cells and just follow up the fluorescence staining, which indicated us if the protein could be secreted. And indeed, once the trophosites start the interaction with epithelial cells, then you can see the fluorescence there decreases. But at 2 hours, what we found is that some of the trophosites did not have any inflorescence anymore, suggesting that the protein has been secreted. And indeed, we could see some prints in the epithelial cells, which were actually stained by the monoclonal antibody. So yes, the Giardia is expressed on the surface, but it's also secreted upon interaction with the epithelial cells. Now we want to know what is that the Giardia does into the epithelial cells. And in order to do that, we use two cell lines, YEC6, and MGCK cells. And when we added the Giardia to these cells, either the purified Giardia or the trophosites expressing there, we then see at 90 and 120 minutes after interaction that the Giardia as well as the trophosites expressing there. Observation then was the fact that there were some cells that can have that look like apoptotic bodies, as I show you here in this picture. If we now look at what were the effects of the Giardia in the MDCK cells that we all know have very tight junction in the monolayer, then indeed it does also have a separation among the cells. And interestingly, there were also holes in them, and you can see them whether you add the purified Giardia or the trophosites. So what then we thought maybe the problem, the fact that the Giardia is affected, Giardia-1 is affecting the epithelial cells may have to deal with the expression with the junctional proteins. And in order to look at that, what we did was to add the Giardia to the epithelial cells and then look for different junctional proteins, and we included ecadherin, occludin, and clodin. And as you can see here, soon after we added the Giardia, we can see that the distribution of these proteins, first of all, that it actually colocalizes with Giardia, and also that at this time there was the colocalization, but in later times what we saw was that the protein Giardia causes the disruption of the pattern and the distribution of occludin on clothing, to a lesser extent to in ecallorine, but at 120 minutes after incubation, then you can see that the monolayer is really very damaged. And indeed, when we looked at the different junctional proteins in a Western blot analysis, we can see that clodin and occludin are the ones that are mainly affected, while ecallorine is not so much affected. And by doing immunoprecipitation assays, what we found was the occludin is the one that actually immunoprecipitates together with giardia, and this immunoprecipitation actually affects also clothing. And as I'm sure you know that when there is a damage in the epithelium there is there is an increase in permeability, then we measure the actual transepithelial resistance in NTCK cells incubated with the Giardia. And as you can see here, there is a time-dependent and concentration-dependent phenomena in the decrease on transepithelial resistance. As the Giardia goes up, then you have the transability on epithelial cell monolayers. So there are two very important observations that we do. Then, when I show you that Giardia induces the apoptotic bodies that we saw in the epithelial cell interactions, then we wanted to know whether that were related to apoptotic effects. And in fact, yes, it does. That we have looked to annexin 5 by FITSI, and then this is a control, this is a non-treated cells. But indeed, when you add Giardia, then you can see that annexin 5 is expressed on the surface, indicating that it is an apoptotic phenomenon. And when you look at the caspase 3 fragment, then you can see that it increases within time. This is a control with actin, and the PAR fragments that also are important in the process of apoptosis. So Giardia indeed induces apoptosis. Next, what we did was, you remember when I mentioned that the inhibitor E64 was very useful for us when we started working on trying to define the proteolytic activity secreted by Giardia. In this case, what we did was we actually exposed the Giardia or inhibit the Giardia with the E64. And here what I show you is that when you inhibit the Giardia with by pre-incubation with E64, you can see that the monolayer is very much like the culture, like the monolayer cell culture, only in medium. Here, as you can see, this is the monolayer exposed to Giardia, and you can see the cells are separated between them. And interestingly, when we use a papain, which is a non-related but a protein from the similar family, there were no reactivity. Here, I just show you that the E64 indeed abolished the proteolytic activity. So, well, if we have already shown that, what would happen if we silence the Giardia gene? And we did that, and we used a very common method for antisense RNA. At the time, we still didn't have all these beautiful methods that are now available that CRISPR has, but it did work very well. We first cloned the Giardia, and then we used the silence method, the transfected trophozoites. Then we did the silence method with antisense RNA, and this is an inducible method with doxycycline. And as you can see here, at 4 hours after induction, the transfected with the PAC Giardia plasmid, and then induction with the doxycycline. And I just wanted to have a look at this part of the panel, in which you can see that there are no damage as compared to the one that is observed with trophozoites that are being that were put into contact with the Yeats's monolayers, and they transfected the W transfected with just the packed plasmid, and here also when no induction. And it's important just to see that indeed if you compare the damage that you see when the trophozoites are expressing the Giardia and the one when they are silenced, we have a decrease in the damage that occurs in the epithelial cells. So we decided then to look at supernatant, not only to look at the damage that the cells were having because of the silence of the gene, but also to the supernatants and look for the proteolytic activity of this the Giardia 1. And in doing so, we find out that the supernatants from the cells that were incubated with the trophozoites that have silence in the Giardia indeed have less proteolytic activity as compared to the ones that are high expression of Giardia or which are only transfected with the only the pack. And these are the ones that are not induced by deoxycycline. So Giardia is a protein that actually causes damage in the epithelial cells, that if you silence the protein, then you'd reduce its activity. Another protein that I mentioned that I was going to talk about was a BSP9B10A protein. This is a variant surface protein. You already listened to Dr. Lujan's presentation where he showed us the importance of these proteins in inducing protection. But here, what we found is that the B10A protein has got a proteolytic activity, different from the one that I've shown you regarding the Giardia, but it also has these proteolytic activities, 2D gels, in which we saw the semogram analysis, and you can see here the proteolytic activity here, the spots corresponding to this protein. So, since as I mentioned that Dr. Lujan referred to variable surface protein, they call the entire surface of the trophozoite and flagella is expressed one at a time, and is spontaneous in vitro replacement, and they were involved in the immune response, the evasion of the immune response, the recurrent infections, and induced protection. So what we wanted to do is to have a constitutively expressed system for this particular BSP, so that then we can actually work with it. And what we did then was to clone this the BSP gene using these vectors, transfect trophozoites. This is the actual schematic representation of this protein. Sorry. So we transfected the trophozoites, and then we selected with this method using puromycin, and then we can actually do the assays that we wanted. So first of all, that is what we wanted to see, is that the VSP, I'm going to refer to VSP for a short nomination, but I will, that refers to the BSP9B110A. So when the trophozoites are expressing this particular protein, while the W trophozoites, there are very few of them. So with this transfected trophozoites, then we can actually go further and try to find out what are the effects that this protein has on the epithelial cells. So here, first of all, is the localization of this protein on the surface. As you can see, also it stains the surface and the flagella, but it interacts. In this case, we actually use CACO2 cells and CACO2 T7 cells, which when the trophozoites interact with the epithelial cells, you can see again that there is a different distribution of the protein, indicating that the interaction of these trophozoites with the epithelial cells have an effect on the localization of this protein. If now then, having the transfected trophozoite with this VSP protein, what we did was detect to make the interaction assays with YEC cells, MDCK, and now with CACO2 cells. And as you can see here, it is just the control cultures in which the cells, the monolayer is normal. If you incubate it with WV trophozoites, then there is perhaps some damage. But in the when you culture the cells with the transfected trophozoites, then you can see again there is quite a lot of damage. Then the cells separate between themselves. Here is YEC cells. Here is the MDCK cells and CACO2 cells. They are gaps in between, and also the monolayer is quite damaged. So this is, I'm just going to show you the localization, this in particular of Z01, which is another protein of the intracellular cells, and the cytoplasmic localization of C01 in CACO2 cells and the transfected trophozoites. It actually localizes from the cell periphery to the cytoplasm, and that is one of the effects that it has in the epithelial cells. I'm not going to devote more time in other experiments that we are currently doing, but indeed it shows us that again this BSP9B1 protein interacts. We have shown that these two cysteine proteases, that the Giardia and the VSP have proteolytic activity, and that they do have the effect on the epithelial cells. However, it was very interesting for us to find out if we use a model, an experimental model, and in this case we use Goebbels, to study the effect of Giardia 1 and the transfected trophozoites in the duodenal epithelium. And in order to use the Giardia 1, which is a protein, as I mentioned before, what we decided to use is the model of ligated loop, duodenal ligated loop in Goebbels. And then we can inject, while we do the loop within the animal, we inject directly the Giardia. And on the other hand, we infected the Goebbels with trophozoites which are expressing the Giardia. I'm going to refer mainly to our work, or the work that we've done with Giardia as opposed to VSP. So once we have the Giardia transfected trophozoites, and we immunized the animals, and then we looked at the different ways to look for the histological sections, staining with hematoxylin eosin. We did immunohistochemistry, and frozen sections with defined antibodies. These will allow us then to see in vivo what could be the effects that trophozoites that are expressing particularly this protein will actually do. And I think I emphasize that because, yes, there are a lot of other groups, but there are several groups, I should say, that have worked with it. But in this particular case, we have a population of Giardia that we can define by what it is expressing. And in this case, it's expressing Giardia. So I'm just showing here that Giardia induces apoptosis in duodenal epithelial cells, and this was done by tunnel assay. This is just the control that you see, of course, this little spots that show you that there's cells in apoptosis. But here, when you use the purified Giardia after 6 hours for infection, you can see that indeed there is apoptosis being induced in the duodenal intestine, the duodenal tissue in the intestinal loop. So these reminds us actually to some of the work that was published some time ago by Troeger and colleagues, with which they show that the patients with Giardiasis also have apoptotic cells. This is the control series is with no Giardiasis, and here is the thin section of a human duodenal specimen, and they use basically the same tunnel assay, and they show that indeed there are apoptosis in cells. So it just reminds us that that also occurs in humans. So then we cloned, as I mentioned, the Giardia gene, basically following the same protocol that I mentioned before. And just for those of you which are not familiar, and particularly for the students that they feel enthusiastic about looking at what can we see in the animal model. And this is for some of you that may not be too familiar with the morphology of the duodenal mucosa. Here we have the villi, this is the lumen, and this is the mucous membrane, two mucosa and spheroids. And this is, I had to mention that it's hematoxylin and eosin staining. And this is the brush border and a simple columnar epithelial nucleus that are very well aligned in the villi, intraepithelial lymphocytes. Work that is in progress, and I decided to include it here because it's important that you can see that when you do have a tool, and that tool can be used in vivo, then that helps a lot to understand what is doing this particular protein. So what we are looking in here is when we transfected trophozoites induce damage in the duodenum of infecting gravels. And here is the control. And as I showed you before, there's a normal architecture of the epithelium. However, if you infect the animals with a WB strain that is expressing Giardia, then you see that there are some changes, particularly there is cellular infiltrate. You can see it in here, and there is some damage to the microvilli. There is a loss of brush border, and also there is severe stratified epithelium. And as you probably can see here, we got a little bit more damage, or more damage, I should say, when the transfected trophozoites are used for the infection of the animal. Then, again, I go back to the work of Troger and colleagues, and as you can see here, it's a section from a patient with chronic giardiasis. It's a biopsy, and then you can see that infiltrate also is here. And I want to just call your attention to these cells here, and you will see it. This is a normal architecture. So yes, the germs are different in terms of in terms of the morphology, but indeed here you can see that in the humans previously reported are these sort of changes. Now, I mentioned that you have to keep an eye on those cells, and these are the globate cells. Globate cells are very important and in producing mucus. And what we see here again, this is the control, which was just given PDS, and then it's a 20 days, 14 days. You can see that there are far more hyperplasia and hypertrophy of globate cells. There are lots of cells in the villi, and you see them here and here, as opposed to in the normal cell, in the normal of the control animals. Here is the hyperpopulation hypertrophy. There's lots of cells, and importantly, the distribution of the cells in the animals, in the normal animals, in the animals given just PDS. You can see that the cells are all along the villi. Here, you can see the cells accumulate in the crypti of the cells, and some of them. And here is even more pronounced that in the crypti and in some of the villi, in fact, are the infiltrate. As I mentioned, it's a feature of this particular. I just want to point out at 40 days there are still trophozoites on the duodenum. At 21 days there are no trophozoites anymore, but still you have this very pronounced effect on the globate cell distribution. And when we looked at mucus, and in fact we actually detect MUC2, which is one of the mucins which are very prevalent in the mucins in the mucus, in the intestinal epithelial, then we did this by using an antibody against this protein, or glycoprotein, I should say. And here's again WD trophocytes, and here is with the transfected trophocytes. And you can see here that the mucus has got a distribution, normal distribution that we can see. While at 21 days, sorry, in the PBS, here is a normal distribution at 14 and 21 days. But when we looked at the distribution of mucus, there is a loss of mucus layer. We cannot see it here as we see it in here, but there is also a MUC2 accumulation, as you can see. So this protein, the Giardia, induces this production of mucus, and it accumulates in the epithelium. So so I just wanted to call an attention. The model I show you is an in vivo model in which you can actually see what is the effect of this particular Giardia. However, all the groups have also looked at the effects of these proteases in Giardia, and in a very elegant work by the group of Dr. André Buret in Calgary University, they use human microbiota films, which they can actually expose to Giardia. And what they found, what I think was quite interesting, is that the exposure to Giardia, or the exposure to the human microbiota biofilms to Giardia, causes the biofilms, and here is when you culture it with Giardia. And then what they did was to look at the thickness of the microbiota films, and they cultured the microbiota. First, they looked at the thickness to find out whether the Giardia can affect this. But in this case, instead of using the trophocytes, what they use is a media in which they have cultured Giardia, and that media contains proteases. So what they did was to add this, as they call the medium in which the Giardia were grown, and if they add it, then they see that there is a thickness in the microbiota biofilms. This is a staining that they did with fluorescent staining with WGA, or to stain, sorry, WJ, so that if they then incubate the spent medium with this marvelous incubator, at least it has helped us a lot to understand the effect of proteases, then they see that it increases the thickness that you see with with adding this spent medium. If they, as I said, if they incubated, then again they see that the effect is inhibited. So proteases actually act in both to increase the mucus, to prevent mucus to be expelled. That is in vivo, and here is in human microbiota biofilms. And interestingly, what they also did was they cultured the microfilms with the human microfilms with Giardia, and then they take the microbiota from these biofilms, and then they colonize the intestine of germ-free mice. And what they found is that Giardia changes the composition of bacterial communities within when they interact with the biofilms. And here you see with no exposure to Giardia, and here are the bacteria, the results when the animals were given bacteria from the biofilms that were exposed to Giardia. And as you can see, there are changes, and here are are the villi that are they show that we're changing. And then again, the treatment, once the treatment with Giardia increases the some of the bacteria. So what they show with this experiment that Giardia also induces these biosis. And I'm bringing it out because that is also important. We in our in vivo model we're looking at it, but here I thought it was important to include one of the very important components. So in summary, what I have shown you is that Giardia is a cysteine protease released during the interaction of Giardia duodenalis trophocytes with epithelial cells, and this was identified by proteomic analysis, and that Giardia 1, when it's exposed, when it's added to the epithelial cell monolayers, there's membrane labeling, phosphatidine exposure, and the other membrane indicating there's an apoptotic process. Giardia colocalizes with occludin, inducing delocalization and degradation of occludin and collodion. And in the case of MDCK cells, it leads to the appearance of pore-like regions, and the epithelial cell displays some gaps along the cell junction. They decrease the epithelial resistance, there is caspase-3 activity, and of course, there is the hallmarks for apoptosis. And when we silence the Giardia gene in transfected trophocytes, that reduce the Giardia proteases activity and, of course, the damage to cells. In the model that I show you, an in vivo model of the duodenal Guadalupe, we show that it induces apoptosis. And when animals were infected with these transfected trophocytes with the Giardia gene, we saw that it induces inflammatory infiltrate, brush border damage, an irregular villi architecture, decreased macro layer in the villi, and there's accumulation of the nuke cells. So all those results together, well, point out that indeed the Giardia 1, the Giardia 1 is the Giardia virulence factor that plays a role in the pathogenesis of giardiasis. And in relation to the BSP 9B10 or 9B10A protein, it is present on the secretomes, as I show you. It displays proteolytic activity in the trophocytes monolayer interface. It causes also cytotoxic damage in epithelial cells, and they contribute. This protein contributes to alter the correct assembly of SOS protein, SOS1 protein in the apical junction complex, thereby affecting the cellular junction. So again, this protein can also be considered as a virulence factor. However, this is a very, very, how would I call it, difficult slide to read, but very nicely done. Why? Because you got not only the the virulence factors that I mentioned and that I talked to today, but there are others, of course, that we are working in analase. There are other words that are working with ADI and OECT, and they are very much involved with the arginine metabolism, and as well as I was telling you, tenasins as well are other proteins that are the SPF. This is one of the toxins. Sorry, but again, here is something that is very, very important for us: is how all these different, if I call it virulence factor, interact with the immune system. I show you some of the results from the group of Professor André Buret that Giardia causes viruses. However, we have to look and see what are those virulence factors do. We got, and I think there's the group of Dr. Stephen Singer, who has worked very nicely with the immune response against this parasite, and there is the IL-17 is a very important cytokine that participates in the regulation of the immune response, but there are also other cytokines that are present, and that occurs, and it depends on CD8 T cells. Also, there's mast cells, which are very important in the innate immune response, the granulation, and this involved with muscle contraction and exercises. So also it looks complicated. I think we have to look at what is happening in the intersection between the immune response, microbiota, and pathogenesis. I'll show you some of the virulence factors that participate in pathogenesis, of course derived from the trophozoites, but also the microbiota, and indeed the immune response are very important in order to understand the pathogenesis of this important disease, which is giardiasis. So what I think we have to look at for further consideration is in innate responses, which virulence factors from Giardia may be recognized by pattern recognition receptors to activate macrophages and dendritic cells. Do virulent factors induce or suppress differentiation of IL-17 producing CD4 cells, which are needed for control of infection? And also, I think it's important to analyze in more detail if virulence factors are related to the activation of macrophage CD8 T cells that can lead to microvilli shortening and disaccharidase activity. How do secrete before, and whether the virulence factors are feasible candidates for vaccine or drug development. So I want to acknowledge people that are responsible for most of the work that I presented today. This is students, Rodrigo Quesada, one of my colleagues of many times, Rocio Fonseca. These are Diana Chavez, who is also in the Department of Infectometry in Simba, and Ariana Cabrera and Jessica Vasquez as well. Arturo, who's actually helping us a lot with the bioinformatics essays, and Raúl Arguello, who is also a collaborator for quite some time. We also have collaboration from the Faculty of Medicine and also from the Hospital Infantil de México and the Instituto de Investigaciones Médicas, and this is the financial support. And before I finished, I want to share with you something that makes me really, in a way, how would I say, we were losing an entire generation of scientists, the COVID-19 and economic tool. And there is a very nice, there's a very nice, and I'm really sad because it is affecting us a lot. In Mexico, we have a lot of problems. And what I want is to persuade the students and the scientists in our countries to get together with the authorities to actually make them, to actually tell them how important it is to carry on doing science in our countries. So I share with you, you can hear of this link, and I think that's all for me. And thank you very much. Sorry.
Professor Guadalupe?
Yes.
Thank you very, very much for this beautiful presentation and for the also very humane testimony you just gave us. So we really do appreciate all your work and the beautiful presentation, beautiful work. We do have several. Is that okay?
Yes. Could I just finish the presentation so that I can see you?
Yeah, go ahead. You can just end the presentation.
Here we are. Can you see me now?
Yeah. Enough, and there is a way for you to interrupt the presentation. But then, so just start presentando by the bottom right. You can click that. Yeah.
Okay.
Yeah.
Thank you.
Now I guess you can see everyone, and I can see you.
Thank you.
Is it good?
Yes, I can see. I can hear you, but sometimes it's like not continuous. I don't know whether it's the system or what it is.
Yeah, maybe my connection is not the best right now. I'll try to speak slowly, and if you don't understand me, just let me know, and I'll try to repeat myself. So, some people were asking about the Japan. So, for example, Professor Vanderlei Souza was asking how is the protease released from proposal ice? Is there a microvesicle or ectosome involved? And Professor José Reis was asking a similar question on the same direction.
I'm sorry, Fabio, I cannot understand very well. Would you mind?
Sure. Yeah. So, can you hear me? Is it better?
I can hear you, but somehow it seems like you are very close to the microphone. I don't know whether that is the problem.
Let's try to change one thing here, and
Yes,
we'll see if that's better. Just one second.
Yes, I think.
Is it better now?
Yes, much better. Thank you.
Okay. Yeah, it's just my regular microphone is not the best.
Okay.
Okay. So Professor Vanderlei de Souza was asking about the secretion of the protease. He was asking, is it like microvesicles involved? And Professor José Reis was also asking the same question. So a very similar question. So could you talk a little bit more about that?
Well, we don't know, Wanderlei, how these proteins are secreted. We started a project in a few months in order to look for the secretion or how these proteins are secreted. But at the moment, we don't know how they are secreted. But maybe they can be involved in exosomes. We don't know.
I see. And Vanderlei as well was asking if you look for any change of junctional proteins in infected gerbials.
Yes, we did. The one thing, Wanderlei, there is intestinal loop model, and we did find, but it's very difficult to actually detect them because it occurs very quickly. And if the intestine repairs itself very quickly as well. So it's difficult to actually visualize that. But yes, we tried.
I see. Professor Reich was also asking about the Giardia's VSGs, and he was specifically asking if they are related to the trypanosomas VSGs. Is there any homology?
I really haven't looked whether it's homology, but I don't remember from reading the work of Dr. Lujan's group and also from Ted Nash group and Frangilin in some time ago. I don't remember whether there are the similarities. It looks to me that no, there are. And again, he explained the way that VSP are actually expressed one at a time with the dicer and the model. Do you remember? So that I think is a bit different.
I see. José dos Santos was asking if you checked whether Giardipain induces mesenchymal epithelial transition. Is there any correlation that you know of between giardiasis and cancer?
No, we haven't checked that yet. We may be looking at the mesenchymal there, but I know there are some groups that have claimed that yes, Giardia can induce cancer. But no, we haven't checked that.
Okay. And a question from myself also, do you know anything about the involvement of the adult stem cell niche with the virulence or with the repair following Giardia infection?
You mean the stem cells?
Yeah, the adult stem cell niche.
Adult stem. No, we haven't looked at that. I don't know of any group that have already, but it's a good idea.
Okay. Yeah, I mean, by adult, I meant intestinal stem cell niche, but yeah, sorry. Okay, so Victor was asking if you believe that protease, Victor Middle, as a researcher from Fiocruz, if you believe that protease may have a direct link to the modulation of microbiota.
That's a question that we all will want to ask, to answer, actually. I think we got the tools to prove that, to see whether, yes, it does happen. That's what I mentioned at the beginning, that the transfected phosphorylase are sure that at least that protein is being expressed. And I forgot to mention, but if you take days after the infection, so we are very much looking at if these proteins are involved in, of course, microbiota dysbiosis. Yes.
Yes. And Hosiani was asking about your experiments with the gerbils, if you control the microbiota in any way.
No, we didn't. That was just, they are not SPF animals. They are normal animals. Yes.
Okay. And also, I mean, it's a question from myself, and I guess it's pretty much the same answer, but I will try it again. Do you have an idea of what would happen if you're trying to use germ-free mice or germ-free systems in these gerbil models? Do you think you get the same results, inflammation-wise, cell death-wise?
Based on what the group of André Buret has shown, that the biofilms, you can incubate those with Giardia, and it changes the bacteria. So I guess that maybe you wouldn't have an infection, because microbiota is very important.
Mm-hmm. I see. Okay. So it's still on the microbiota. So do you know how would this signaling? Sorry, just let me rephrase it. Signaling change.
No.
Yeah. Okay. Patients with genetic disorders where there is less secretion of mucins. Do you know anything about that?
We, with patients, haven't looked really much into it, and from the literature, I don't remember that there are more severe of genetic disorders, but one has to look at more carefully, I guess, especially in our countries in which Giardia is endemic. So we can actually have a look. Maybe there are some reports, but I haven't come across them.
Okay. And Ana Gadelha was asking if you could comment on the use of, or the potential use of Giardia as a therapeutical target.
Well, we are actually addressing that. I'm glad all these questions come from very nice and thoughtful persons. Yes, we are addressing that. I don't have the answer now, but yes, we're addressing that.
We'll be looking forward to that. It's very exciting. So another professor was asking if you could comment a little bit more on the technical side about the best conditions for the establishment of the parasites in terms of quantity of intestinal mucus. So
one of the non-specific barriers for for the invasion of any pathogen within the intestine. However, if you looked at some of our results in the germ model, that it seems that the dysfunction of the epithelial intestinal epithelium prevents that mucus to be functioning the best way, right? So yes, it does, because we have seen it in many other pathogens, and it is a barrier, which is important. But then it may be that it depends on the time in which you actually look at. You look very early, which we have done. I just didn't have the time to show the results. Then there is mucus that is displayed in such a way that is when the trophophorites are. But then afterwards, the mucus seems to be the expulsion of the mucus seems to be a bit different. Maybe this is because of the damage that has already occurred within the epithelium.
I see. And Sophia Aguillo was asking about the mechanism of cell death is it apoptosis? Is it any other kind of cell death? Did you try to look at that into more details?
We basically, with the Giardia is apoptosis, so we have actually checked with different other means whether there are other cell death patterns. No. With other proteins that we are studying, like amylase, yes, we see that there is a different kind of cell death.
Okay. Do you know anything about that?
Well, occludin immunoprecipitates with in our system, immunoprecipitates with Giardia. So it may be that there are some receptors there that actually that helps them to localize where occludin is. But that's basically what we have done.
Okay. We have one more question, and maybe if someone here on Google Meet would like to address a question himself to Professor Guadalupe, we might have some time to do so. But I mean, one final question that I have from my list is actually, we would like to ask you a favor almost. It's not so much of a question, but could you give us some kind of advice or some kind of message to all students? It's such a time right now where science, especially in countries like us and like Brazil and Mexico, might be so undervalued.
Yes, I will. Certainly.
We will be very excited for that.
Well, you want me to do it now?
Yes, please.
Okay. No, not really. What I want to say is, believe me, you are the most important part of our groups. You are the thoughtful as well as the people that contribute to our research projects. And please, you have to feel like you can do it. We as lecturers and as professors within the department are always happy to help you. How? Supporting you, giving not only that, but to make you sure that you can actually pursue the projects that you propose. In our countries, as I say, it is very difficult. But when you read, once you read the paper that was published in Centro, you will see that if we get together, as we were talking before the presentation, if we get together with the students, talk to them, and we go together again with the authorities to tell them how important it is that they keep actually supporting us, then we can manage. Also, I think you got very young scientists that I met in one of the meetings in Giardia, and they're very good. So from me, anything that I can help them, you're more than welcome. But more than that, I pursue you to carry on with your studies. And if you have anything to offer to us as a group in that work on Giardia, please do contact us, talk to us. We are more than welcome. You're going to be more than welcome, since you are the best part of the science.
Amazing. Professor Guadalupe, once again, I would just like to thank you very much for this beautiful presentation, for your very kind words and your very humane testimony. I will now pass to Professor Susana Frases, and she'll start wrapping up this presentation.
Thank you, Fabian. That's very, very kind of you.
Muitíssimo obrigada. Se a senhora me permite, vou esperar que todos nossos alunos entendessem o que a professora Guadalupe falou. Ela falou, acho que uma coisa muito importante, o futuro da ciência está com vocês. Acho que das 47 pessoas ainda logadas, a maior parte ainda são alunos. Então, vocês estão escutando isso no meu portunhol exquisito, em inglês, em espanhol e em todos os idiomas. Então, a ciência é com vocês, meninos, e as dificuldades a gente luta junto. Muchas gracias, Guadalupe, por tus palabras de aliento y de esperanza para nuestros alumnos. Nosotros en el día de hoy es importantísimo. Pero yo creo que de su charla, la parte más importante es ese apoyo que la señora mostró para todos nuestros alumnos, para la ciencia de nuestros países, y cuando falo de nuestros países yo me refiero a Latinoamérica, que mi corazón está aquí, aunque yo nací allí. España no es muy diferente, porque tampoco se valoriza la ciencia ni se da importancia a los científicos de allí. Entonces yo creo que el mundo latinoamericano es todo uno, y con seguridad, luchando juntos, llegaremos a algún lugar. Cada uno, nadie solo en la vida consigue nada. Entonces, muchísimas gracias. Ha sido un placer conocer a la señora, aunque fuese esa hora y media. Da para ver la maravillosa persona y científica que la señora es. Muchísimas obrigada en nombre de la posgraduación en Ciencias Biológicas Biofísica, y en nombre del Instituto de Biofísica, y en el nombre de la Universidad Federal de Río de Janeiro. Muchísimas obrigada. Muchas gracias. Thank you very much for your talk and your words.
Thank you.
Vanderlei.
Yo voy a hablar en portuñol. Yo creo que la palestra de Guadalupe mostró el lado de la ciencia y el lado de la pasión. Pasión por la ciencia, pero también la pasión que se transforma en revuelta contra nuestras autoridades, que colocan, aunque dicen que ciencia es fundamental, y la cuestión de la COVID, estão sempre hablando que ahora es el momento de la ciencia, es el momento de la ciencia, pero esto se queda como una retórica que no se traduce en fatos.
Cierto.
Hace 2 años yo tuve la oportunidad de ir a México para receber el premio de la Fundación Miguel Alemán. En aquella oportunidad tive el contacto con la nueva ministra que iría a cuidar de la ciencia, de la educación, la nueva presidente del CONACYT. Estávamos todos muy entusiasmados en tener un nuevo tipo de apoyo para la ciencia y la cooperación de Brasil y México. Pero nada se pasó por los varios motivos. Quer dizer, lo que se ve es que no hubo en la realidad ningún esfuerzo por parte de las autoridades mexicanas y brasileñas de incrementar la natural cooperación que debería existir entre Brasil y México como dos naciones centrales de la América Latina. Gracias, Guadalupe, que continuemos trabajando apesar de tudo, y que nos encontremos muy brevemente en el próximo congreso de Giardias ou de Giardia e criptosporídeo, que já não sei quando será, em que oportunidade, e assim. E eu diria que tivemos 150 pessoas participando entre o Google Meet e o YouTube. Certamente, como a palestra vai se quedar no YouTube, muitas outras pessoas terão a oportunidade de poder ouvir-te. Gracias y hasta pronto.
Muchas gracias a todos. Realmente fue un placer estar con ustedes. Hasta luego.
Gracias.
Muchas gracias, profesora.
Muchas gracias, profesora.
Muchas gracias, Fabio. Y mis saludos a Marlene, por favor. Gracias.